Hi, it's Brett the Lab Tech, and I'm here in the lab today to run through the photosynthesis lab, particularly the experiment where we're testing for starch. So I'm working in the fume hood today because the first step in this process is to boil the leaf in alcohol, and I don't want to be breathing those fumes. And this is going to extract the pigments out of the leaf so that we can see the starch test a little bit more clearly. Now, boiling alcohol is a little bit dangerous. We don't want to boil the alcohol directly on the hot plate, so we're going to set up a double boiler. So we'll have a small beaker with alcohol inside a larger beaker that has just tap water with some of these boiling chips inside. So we'll put the tap water and boiling chips back on the hot plate. Get some ethanol. And we're using 80% ethanol. And we'll put the smaller beaker inside the larger beaker. And we'll turn the hot plate up to high. Okay. We have that getting as hot as it can, as quickly as it can. And while we're waiting, we'll take a look at the leaf that we're going to use. Last week I set up this geranium plant with these 4 different filters on it. So we have green, red, blue, and then a piece of black electrical tape to block all the light. So the color of the filter is the color of light that is getting to the leaf. And I set up a few of these just in case we need to run this experiment again. And before we put the leaf in the alcohol, we're going to want to sketch the positions of these filters. And before we even do that, I'm going to mark this leaf so that we know what the orientation is. And the way I'm going to mark it is just to punch a hole in the other side of the leaf. Okay, so this will kind of help us remain oriented, and we'll know that the filters are on the opposite side of the leaf from the punch. Okay, I'm going to peel this leaf off now, and you can sketch those filters. And then I'm going to go ahead and pause the video while we wait for the alcohol to warm up. All right, so the water in our double boiler is starting to boil. It's kind of at a gentle boil. The alcohol is maybe not quite boiling yet, but I'm going to go ahead and turn this down to about halfway because I don't want the boil to be too violent. Okay. Now I'm going to take the filters off the leaf before they go in the alcohol. And we've already sketched these, right? So we remember where all the filters are. Try and do this carefully without ripping up the leaf. Oh, took a little piece off of it, but still pretty much intact. And sometimes you can see an outline of where some of those filters were, but we can't really with this leaf. And we will go ahead and put it into the beaker of alcohol and try and make sure I'm going to make sure the whole leaf is submerged. It's okay if the stem isn't. And we're going to let this boil for a while. And as it boils, the pigment will extract out of the leaf and into the alcohol. which is going to help us see the starch when we test for that. So once again, I'm not going to make you watch the leaf boil for, I don't know, 5 minutes or so. I'll pause the video and come back when the leaf is pretty much done. Okay, it's been maybe about 5 minutes, and the leaf is pretty well bleached out by now. As you can see, this is what the leaf looks like after boiling. And yeah, I think it's ready to do the starch experiment. I'm going to go ahead and turn off the hot plate. It's going to stay warm for a while, and after the alcohol cools down, I'll put that in the waste container. But we'll just let that sit while we move to the next spot, or next step. What I'm going to do here is just rinse the leaf with some alcohol, or rinse the alcohol off the leaf with some deionized water. Excuse me. And kind of gently spread the leaf out. You can see where the hole punch is. And if we remember, those filters were on the opposite side from the hole punch. So I'm going to be careful. The leaf gets really fragile after you boil it. So just be pretty careful with that. Okay, so now we're ready for the next step. I'm going to go ahead and take the rinse water and dump that out in the sink, and then show you the staining step. Okay, so now we're ready to stain this leaf. And to stain for starch, we're going to use 20% Lugols iodine. Iodine and starch react, and we'll see that reaction here. And the key with this is not to let it stain too much. So if we leave this iodine on there, it's just going to keep staining and staining and staining. So at some point we'll pour off the iodine and rinse it with deionized water to halt the staining. And the key to this is to just kind of flood the leaf with iodine. It'll probably take several droppers full to do this. I'm probably most concerned with the area where the filters were, which was around in here. So we'll let this go for a little bit. Okay. Can you gently slosh the iodine around a little bit? And you can see the unfiltered parts of the leaf are staining pretty dark, and we're starting to see the outline of where the filters were. I'm going to let this go just a little bit longer, and then I'll pour off the iodine and rinse it off. that might be about as stained as it's going to get. I'm going to go ahead and pour that off, then come back and rinse it. And again, just rinsing with some deionized water. The staining process stops. And pour that off. I might do that a few times just to okay. So now we have our stained and rinsed leaf, and it's time to read the results. I'm actually going to move this over to a light box so we can maybe see it a little bit better, although we can see it pretty well here, right? We had the area with the black filter paper, or the black electrical tape, and the different colors here. But we can maybe see this a little bit better on the light box. Okay, so here are the results. As you can see, most of the leaf that was unfiltered stained fairly dark. There's that black filter. This is the same orientation of the filter, and you can kind of see the areas where the blue filter was and where the green filter was, and in between, maybe a little bit more starch. But overall, this part of the leaf didn't have as much starch produced. Okay, so I'm going to let you interpret those results, and that concludes this experiment.